So gluconeogenesis, it means we synthesize new glucose. So it's also called GNG. So this is glucose synthesis. So the basic idea here is we're going to use pyruvate lactic acid, some Krebs cycle intermediates, and some amino acids, actually, are glucogenic. Some are ketogenic, some are glucogenic. We'll talk about what that means. And so we start with these products and then we make glucose. This is similar to reverse glycolysis. But we bypass some steps. We'll talk about that. And like those steps we bypass require different enzymes. So now I'm going to write out the names of the things instead of just 6-carbon or whatever. So let's do it this way. Yeah, so let's start with glucose. The first thing we do is we stick a phosphate on it, right? So we get glucose 6-phosphate. Then we isomerize it, so we get fructose 6-phosphate. Then we stick another phosphate onto that, so we get fructose 1,6-bisphosphate. Then we go through a couple of reactions and we get something called phosphoenol pyruvate. Which is PEP, and then after that we get pyruvate. So remember, we get 2 of these and 2 of these. Okay, so I'm going to put glycolysis over here and gluconeogenesis over here. So the idea is we do glycolysis when we have a lot of glucose and not a lot of ATP. We do gluconeogenesis when we have not so much glucose, but we have stored ATP. So like the idea is like, think of ancestral human beings. We'd have like a big meal, but before that meal we didn't have anything for like a couple days.
Yeah.
So we would have had low ATP, so we eat the big meal, and now we have high glucose, and we can do this.
Yeah.
But then a day or 2 passes by, so we have the stored ATP from that, and now we have low glucose, so we get to make some.
Yeah.
Okay, so that's the idea. So in glycolysis, right, glucose, we know this is hexokinase. And then here is phosphofructokinase. I'm just going to write PFK. Now, pyruvate kinase also has some allosteric regulators. So one of them is phosphoenolpyruvate itself is a positive allosteric regulator. And then fructose 1,6-bisphosphate is going to be a positive allosteric regulator of this thing, right? So once we make this fructose 1,6-bisphosphate, this molecule is committed to glycolysis. So before this step up here, where we have glucose 6-phosphate and fructose 6-phosphate, we can do other things. There are other metabolic pathways these molecules can be a part of, like making nucleotides, for example. But once we go through PFK and make fructose 1,6-bisphosphate, we have to go all the way to pyruvate. So it makes sense then that these would be positive allosteric regulators of pyruvate kinase. And then inhibitors are going to be ATP and the amino acid alanine, which is not that important. Okay, so that is that. I again just want to re-emphasize that the most important positive allosteric regulator of phosphofructokinase is fructose 2,6-bisphosphate. So there are other ones, but I'm just highlighting that one for right now because it's most likely to be tested. Okay, now for gluconeogenesis. So in gluconeogenesis, we're going in the reverse direction, right? We want to get to glucose. So first, we have pyruvate to phosphoenolpyruvate. That doesn't work for us. So we have to bypass that. So we go from pyruvate to the Krebs cycle intermediate, oxaloacetate, or OAA. So this is that 4-carbon thing that combined with acetyl-CoA to make the 6-carbon citrate. So oxaloacetate, right? Remember we have 2, so we get 2 of these. And this process takes 2 ATP and we use the enzyme pyruvate carboxylase. And it's just for completeness, acetyl-CoA is a positive allosteric regulator of this. So if we have a ton of acetyl-CoA around, right, it means we've done too much glycolysis, right? So we're going to make some glucose with the pyruvate that we have instead of metabolize it. And then there's this weird thing. I don't know if this will be studied, but remember this pyruvate we're starting here in the mitochondrial matrix. So this actually happens in the mitochondrial matrix. So this pyruvate has to be shuttled out, or this oxaloacetate has to be shuttled out. So there's this weird thing where it gets converted to malate and then converted back to oxaloacetate. So it gets converted to malate to shuttle it out of the mitochondria and then back to oxaloacetate. I don't know if that's tested, but it could be. So then we have oxaloacetate now in the cytoplasm. Now that becomes PEP via an enzyme called PEP carboxykinase. So phosphoenolpyruvate carboxykinase. So had so we have pyruvate, then we got a phosphopyruvate, so we have a kinase. That gets us there. And how I remember this enzyme is it's PEP-CK. So it's like Pepsi Cola or something. Pepsi Cola, that's just a mnemonic.
Okay.
And then it takes 2 GTP in here also. So this first step to get pyruvate to PEP takes 4 ATP equivalents. 2 ATP to start and then 2 GTP.
Okay.
Yeah. So then we go from PEP to fructose 1,6-bisphosphate. So this is a 2-step process. And that takes 2 ATP. So now we're 6 ATP in the whole. Now here we have the counterpart to phosphofructokinase. So this is called, these enzymes are removing a phosphate group. So a kinase adds a phosphate group. What removes a phosphate group is a?
So it's the, wait, I'm confused on the.
So.
Sorry, I was just like trying to like, systematically go up the ladder, and I was like reading all of it.
Okay.
I think I got lost in the.
No, that's fine. So in terms of like a general enzyme name,
Yeah.
a kinase adds a phosphate.
Yeah.
What removes a phosphate?
I'm not sure. I don't. Do I know this?
You know this.
It takes the phosphate away. Oh my gosh. I'm like blanking. I don't know. Cytokine?
No. A phosphatase. So a kinase adds a phosphate. A phosphatase removes a phosphate.
Phosphatase.
Okay. So just a general naming thing there. So now we have fructose 1,6-bisphosphate going to fructose 6-phosphate. So we're removing that phosphate that's on this one carbon here.
Yeah.
So we're going to call that fructose 1,6-bisphosphatase.
Okay.
That's the enzyme. So it's the same, it's almost the same as the name of the molecule. But instead of phosphate, it's just phosphatase. So fructose 1,6-P-ACE. So that's how it's abbreviated. F16-P-ACE. And that's sort of the opposite of phosphofructokinase. So it has opposing regulation. So in this case then, citrate is going to be a positive allosteric regulator of this, whereas over here, citrate was a negative allosteric regulator. Similarly, over here, AMP is going to be a negative allosteric regulator, whereas over here, AMP was a positive allosteric regulator. So we have the same molecules with opposing regulation on the flux through the pathway. So that's an important concept because when you look at it on the board, you think like, oh, I'm either doing one or the other.
Yeah.
But in a biological system, both are happening at the same time. So you have so it's these levels, these concentrations of citrate and AMP that are sort of determining the base flux through the pathway. Now, fructose 2,6-bisphosphate, that sort of overrides all of it. But in general, the concept of reciprocal regulation is important here.
Okay.
So the same molecule regulating counter-opposing enzymes allows like one side to be favored over the other. Okay, cool. So that happens, and now here's the key. So we have an isomerase, and then this is also super key. the enzyme going from glucose 6-phosphate to glucose. Super important because it's only in the liver, this enzyme. And so this is going to be, it's going to be the same naming as here, so it's just the name of the molecule, ACE. So glucose 6-phosphatase, G6P ACE. The key thing about this is it's only in the liver. Only in liver. So the biological context of this is that when glucose enters the cell, you immediately stick a phosphate on it because that keeps the glucose in the cell. When this phosphate is removed, the glucose can leave the cell.
Yeah.
Glucose only leaves liver cells, hepatocytes, because the liver regulates blood glucose. So any other cell that's doing gluconeogenesis is going to keep the glucose that it makes.
Okay.
It's going to keep the glucose 6-phosphate that it makes. But the important one is the liver. When we're talking about metabolism, when we're talking about all these metabolic pathways, the important ones are the hepatocytes. I mean, it's happening in every cell. It's capable of doing it. But the hepatocytes are where it really matters a lot. Red blood cells, it also matters, and we'll talk about that at the end. So, so this really matters because it's only in the liver, and so then the liver can release the glucose, and then the glucose can get to the brain or wherever else it needs to go. So we talked about reciprocal regulators. We said citrate was a reciprocal regulator. It's a negative allosteric regulator of phosphofructokinase, whereas the positive allosteric regulator of fructose 1,6-phosphatase. And then same with AMP. AMP is negative here and positive here. So that's the concept of reciprocal regulation. Again, just want to emphasize that.